Hello students, this is Faizan Mirza. We are discussing transport in plants. The last part that we have to cover is phloem and how translocation occurs through phloem. So before we start off with what phloem is and how it's carrying out the process of translocation, we just need to look into the details of the features of the cells which make up the phloem in itself. So in phloem, there is a sieve element and a companion cell, one for one. So if there is a sieve element, there will be a companion cell for it. The companion cell, as the name suggests, is a companion for the sieve element. So sieve element is the main cell through which the transportation will be occurring of the sucrose and the assimilates. And the companion cell will provide all the necessary companionship which is required for this process. Now, what's happening here? The sieve element is having limited cytoplasm, which is pushed to a side. It will have the endoplasmic reticulums, the mitochondria, and the cytoplasms, and the cell surface membrane, and everything which is a part of a living plant cell. But it will be carrying out very limited metabolic activities. The metabolic activities will be just restricted to a very small level. But a companion cell is having all the parts which a typical plant cell is known to be having. It will have a large vacuole, which you can see it's missing here. And this companion cell will be the one which will be allowing sucrose to be transported into the sieve element. So then the sieve element can take the sucrose from one part of the plant to the other. The sieve plates are joined together to form a hollow tube, not exactly as hollow as xylem. This is having cross walls. And these cross walls are the sieve plates. These sieve plates have sieve pores, and these sieve pores have the opening, which actually is the plasmodesmata. And then there are plasmodesmata between a sieve element and a companion cell. And a companion cell will have sieve element, the plasmodesmata with the cells surrounding that companion cell. So, what's happening here? Let's start discussion of how this is happening, what's happening in phloem. So the phloem is involved in the transport of sugars, the assimilates, which could be sugars and the amino acids primarily. These are called assimilates, and these assimilates are transported actively. These assimilates in the phloem sieve element, they dissolve in water, and water provides the hydrostatic pressure for the flow to occur. When the phloem is allowing this transportation to occur, these assimilates can also be termed as phloem sap. And the movement of the phloem sap along the sieve elements in the phloem is called as translocation. Translocation is an active process. Unlike the movement of water in xylem, which is primarily passive, it is not governed by, the water transport is not governed by ATP at all, or even not to a large extent. In xylem, there is no ATP involved. But in translocation, ATP is involved. Without ATP, without the energy from ATP, translocation cannot occur, which means this phloem sieve element cannot transport the assimilates without the presence of or the use of ATP. What's the use of ATP? It's not occurring directly in the sieve element, but in the companion cell, we'll come to it. Aphids. What are aphids? Aphids are insects that feed on a dash, and you can just fill out these dashes yourself. So what's happening here? This is you can see a plant stem, and this is an aphid. It inserts its stylet into this stem, and in the stylet, the mouth part, this stylet goes deep into the vascular bundle, and in the phloem it starts to feed onto the phloem sap. The phloem sap comes out, and it feeds on it, and the callose will not form because the phloem going out is going out at too slow rate. So you can provide this aphid with anesthesia, and after giving anesthesia, the aphid can be removed. Phloem sap still exudes from the broken stylet. You provide anesthesia to make sure that you do this as humanly as possible. Okay, so why do it? That's another question. So yes, why do you do this? You do this because you want the phloem sap to come out, either for you want to just carry on some research activities on the phloem sap, because if you were to take out this phloem sap yourself, the plant would get the stimulus that the phloem sap is getting lost at a very high rate, and it would form callose at the nearest these sieve plates. The sieve pores will be blocked by callose. So you use the aphid for that. You allow the aphid to go and start feeding on it, and then you just decapitate it. Okay. very dramatic. Now the concept of the source in this thing. What is the source? Source is the region of the plant where sugars are produced during photosynthesis. So these parts are the source. So in the source, the photosynthesis occur, and sink is the region of the plant where the carbohydrates are either stored or used up. If the carbohydrates are stored, they are stored in cortex and root storage organs like tuber and rhizomes. For the sugars being used up, they are used up in growing parts of the plant. For example, buds and roots and fruits and nodes and internodes and leaf. Nodes are the region where the new leaf will grow, and internode is the distance between the two leaf stalks, the petioles, and the new leaf. Yes. So phloem transport is the phloem sap from source to sink in most cases. Or it could be from one sink to another sink as well. But mainly it's from source to sink. So what's happening in source? The processes which are occurring, these are highlighted in pink. This is the diagram taken from Mary Jones. You can see carbon and water. The raw materials from the environment are converted into glucose and fructose, and that turns into sucrose, and then you just allow water to go, and this gets loaded into the sieve tube, and the sieve tube allow mass flow to occur. Mass flow occurs and the sucrose goes to the sink. Once in the sucrose is unloaded. When sucrose is unloaded, water also is drawn out of this sieve tube. So this unloaded sucrose, it's converted back to glucose and fructose. These monosaccharides can be used for in respiration, and CO2 and water can form. And with respiration, the ATP which is made can be used for several vital processes. Or this glucose can be stored as starch, or the glucose can be converted into the beta glucoses, and then cellulose can be made again for growth purpose. So glucose is produced in photosynthesis, transported as sucrose, stored as starch, used as glucose or fructose, monosaccharide, disaccharide, polysaccharide, monosaccharide again. Why the monosaccharide is being used here? Why it is transported as disaccharide? Why is it stored as a polysaccharide? The answers of these lie in the second chapter in biological molecules. How translocation occurs. So to understand how translocation is occurring, you need to understand these three cells which are lying side by side. So when translocation is occurring, translocation occurs in two main stages. The stages include number one, loading of sucrose, and number two, unloading of sucrose. Loading occurs at the source, and sucrose is loaded into the phloem. This occurs by mass flow hypothesis. I'll be outlining this in 9 steps, 1 till 9, which I'll annotate here. And the second is the unloading of sucrose from the phloem, where the sucrose is unloaded in the sink. These are the main the main stages which occur in translocation. So these are the 3 cells here. A leaf cell, which is the mesophyll cell where photosynthesis occurs, a companion cell, which breaks the ATP, which is where exactly ATP is used, and sucrose moves from the companion cell, from the source to the companion cell, and then into the sieve element. How is this happening? There are 9 steps here. Number 1, the mesophyll cells, they carry out photosynthesis, and with that photosynthesis, raw materials are used to turn into sugars. You get glucose, and glucose is converted to fructose, and glucose and fructose are joined together to form sucrose. This region is the cell membrane of the companion cell, and then the cell wall. You can just see here, this is the cell wall of the cell, the leaf cell, the mesophyll cell, and this is the cell wall of the companion cell, and this is the cell membrane of the companion cell. Why have I highlighted the cell membrane of the companion cell? Because this membrane is having certain proteins which we are to discuss, because they are of the prime importance in translocation. So the third step is that there is a protein here, a transmembrane protein here, which is the proton pump, the H+ pump. It is located in the cell surface membrane of the companion cell, and this proton pump, it allows the H+ ions to move out of the cell by active transport. This is breaking the ATP into ATP and PI, and the energy used to move H+ ions out of the cell, and from the cell membrane it goes into the cell wall, which is the adjacent cell walls of the 2 cells right here, at that point. So once the H+ have gone out from this cell by active transport, what will happen? There's another transport protein here. This is a co-transport protein. The co-transport protein allow H+ ions and the sucrose to enter together. What's a co-transporter? A co-transporter is a protein located in the membrane of a cell. A co-transporter allow movement of 2 substances in the same direction. This is what the co-transporter is doing. It allows the movement of H+ ion and sucrose together from the source cell into the companion cell. Once this sucrose and H+ have entered, the H+ will keep on moving around in the same way. It goes out by active transport through this proton pump. It comes back again through the co-transporter, bringing sucrose with it. So this is coupled transport occurring. This is active transport occurring. So this is the active transport which makes the movement of sucrose or the loading active. So if ATP is not present, then this process will not occur and sucrose will not move into the sieve element at all. because the companion cell will not be working. So once the sucrose is moving, is coming in the companion cell, it moves into the sieve element through the plasmodesmata. When sucrose enters into the sieve element, this is just diffusion occurring. So through diffusion, it enters into the sieve element. Once it goes into the sieve element, it reduces the water potential there because sucrose is osmotically active. It lowers the water potential. So once the water potential gets lowered inside the sieve element, so you know that what should happen next? Yes, osmosis. So what follows? This 0.7 is osmosis. So water starts to be pulled into the sieve element just following sucrose. So when water enters into the sieve element, it provides the hydrostatic pressure required to initiate the mass flow. This is exactly at the point where pressure-driven mass flow starts to occur, and with pressure-driven mass flow, the assimilates, which is the sucrose here, together with water, it starts to move ahead in the phloem, crossing these plasmodesmatas or the sieve pores. It keeps moving from the source to sink. When this is the entire process of loading. Now, once the sucrose reach the sink, now what happens in the sink? In sink, unloading occurs. For unloading to occur, the sink must use an enzyme to break sucrose back into the monosaccharides, because sucrose is a disaccharide. It must be broken by the enzyme invertase, which is exactly similar to the sucrase. So invertase breaks sucrose into its monosaccharide components or monomers, the glucose and fructose. So sucrose breaks into glucose and fructose, and sucrose and once broken into glucose and fructose, it moves into the sink. So as the sucrose is removed from the sieve element, the water potential starts to rise up and water starts to move out of the phloem into the surrounding xylem or into the surrounding cells in that region. So water starts to go out as well. When water goes out from that phloem, so what's actually happening? This is the end of translocation. The sink was delivered with the sucrose and water moved out of the phloem as well to the surrounding tissue. So this is what's happening in translocation. That's it from my side. Thank you so much.